Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog.
For the public bringing this program here, and for the AP Biology, are you AP Biology people? We've got databases for you online that you can access from home, Science in Context, Salem Press Science. If you go on to our website and go to the databases, check out Science and Technology, and maybe it'll help you with your homework.
Good evening, then. I'm Yvonne Stapp for Science for the Public, and I welcome you to our spring lecture series. And tonight we thank Joanna Breen and the Friends of the Belmont Public Library for hosting this event, as well as two upcoming lectures that you'll see on your handout. We are very pleased to be here on Earth Day, and happy Earth Day to you. This is the day that commemorates the beginning of the environmental movement in the United States some 45 years ago. I'm finding that hard to believe. In the early days of that movement, decades ago, it was easy in some areas of the mainstream to dismiss environmental concerns as expression of a sort of a fringe group of hippies and the like, but today, environmental issues are very much front and center. We now understand that the world is awash with toxins produced by humans, and the central issue is whether organisms will be able to adapt fast enough to unnatural toxins in a rapidly changing complication of global climate. Some species adapt, others struggle, and still others become extinct. And to learn why some organisms thrive and others don't, we have the excellent fortune to hear from an evolutionary toxicologist, Dr. Emily Monosson. Emily Monosson received her Ph.D. from Cornell University, and in addition to extensive research on familiar toxins, things like PCBs and others, she has investigated an emerging group called nanochemicals, and I hope she'll come back and talk to us about those in the future. Along with her work as a consultant, Dr. Monosson is an adjunct professor at the University of Massachusetts Amherst and an independent scholar at the Ronan Institute. Most important of all, she is deeply committed to making the complexities of environmental toxins accessible to the general public. She's the author of two excellent books on the subject, Evolution in a Toxic World, she'll show it to you, and An Unnatural Evolution. I'd like to point out these two books are published by Island Press, a nonprofit, highly respected press devoted to environmental issues. These books are available in our Minuteman library system, and they are must-reads for anyone concerned with how life has adapted to various toxins and how it might adapt now. And now it is a great pleasure to welcome Emily Monosson.
Oh, thank you for that really nice introduction. And thank you all for being here on this Earth Day. And if I'm asked that I say a few words before I get into the really fascinating, this is a great opportunity for me to talk about two fields that I think are fascinating, which is toxicology and evolution, and putting them together has just been great. As she said, it was so interesting to me that I actually did two books on it. But before I do that, Yvonne asked that I say a little bit about what toxicologists do, because she said, a lot of people don't know what toxicologists do. So basically, toxicologists are people who are fascinated by poisonous chemicals and how these chemicals interact with living systems. And I think a lot of people are probably familiar with toxicologists now, compared to when I went to school, because of shows like CSI and X-Files. I was getting my hair cut the other day, and the woman was like, "What do you do?" And I said, "I'm a toxicologist." And she said, "Ooh, do you work with dead bodies?" And so I said, well, maybe dead mice, rats, and fish or something like that. But, so a lot of the majority of toxicologists are doing environmental toxicology. So we study the effects of chemicals that are out in the environment and how they interact with living systems out there. And then a lot of toxicologists are working to protect human health, looking at the chemicals in food in drugs and consumer products. So you read about a lot of that in the news often, what they find. So in a nutshell, that's basically what toxicologists do and why we do toxicology, because we just sort of like to think about poisonous, toxic chemicals. But what I want to do is just give you a very brief background on some of the things that we think about when we're thinking about toxic chemicals. So one of the first things that we need to think about is absorption. Okay, these are sort of the four basic components that any toxicologist kind of should have running around in their head, no matter what they're doing or what level they're working on. So the first thing is absorption. So how does a chemical get into the living system, whether it's a human body or we're talking about a bacterial cell? So you need to think about, can it get into that? Because if it can't, if it's sitting on a bottle or it's just sitting on the top of your skin and it's not going to get in, it's not a problem. It's not going to be toxic to you, right? So that's the first thing. Absorption. Can the chemical get into the system that we care about? The next thing is what happens when it gets into the system. So does that chemical distribute throughout the system? And where does it go? So if a chemical comes in and then it just goes out, probably not a problem. If it comes in, starts to distribute throughout the bloodstream, might get to your brain, might get to your liver or bones or wherever, then that starts to, help a toxicologist figure out where they should start looking for adverse effects or how that chemical might cause bad consequences. So absorption is the next thing. And then this is the part that I think a lot of toxicologists are working on and think is kind of the most interesting part, is the metabolism. So once a chemical gets in, it distributes throughout a body, then what happens to it? Do you have enzymes and proteins, do they interact with that chemical, and how do they interact with it? So you often hear about things getting metabolized. There's a huge field in toxicology about detoxification. We have a lot of enzymes, proteins, that interact with chemicals that are able to detoxify these chemicals. They either break them apart or they add things to them that make them easier to excrete or to get out of your body. So it's this really amazing detoxification system. And there's a lot of different kinds of detoxification, a lot of different kinds of enzymes that do this. So it's really sort of a focus of a lot of work that people do. And then the last part is that we think about is excretion. So once a chemical comes in, it gets distributed, it might get metabolized, it might get broken down, and then does it get excreted, and in what form does it get excreted? So sometimes they don't get excreted. So when we think about Earth Day, some of the chemicals that really set off Earth Day that Rachel Carson wrote about were those kinds of chemicals that really didn't get excreted very well. They once they distributed in the body, they got into the fat and they sort of stuck there. And they didn't get out of a body, and so that caused a lot of problems, especially when that happened out in the environment. So we think about excretion. So these are sort of the four basic things that toxicologists are thinking about when we're doing our work: absorption, distribution, metabolism, and excretion. And so I did a lot of teaching and I did a lot of writing about this, and eventually one thing that we don't think about often is evolution. And so I started thinking, as I was trying to explain these things to students and to readers, I was thinking, well, it's really cool that we have this detoxification system. We have all these we have skin that can keep chemicals out, and we have all these things. But how did these evolve? So what were the sort of conditions where life evolved some of these amazing kind of protective systems? So this is what got me interested in evolution. And so I'm going to talk about, for the rest of this talk, start talking about evolution and sort of toxicology in an evolutionary context. And before I do that, there are sort of three kind of basic principles for evolution. So before we think about, evolution, we need to think about genetic variation. So for evolution to happen, we need to have genetic variation. If you had one population, within your population, no variation, you can't have evolution happen. The next thing is that variation has to be heritable. You have to be able to inherit any kinds of changes. Okay, so if it's not heritable, if you, evolve some kind of cool third hand that can, you're born with some kind of cool hand that can do great things and it gives you a great advantage, well, if you can't pass that off to your offspring, it's not going to matter in an evolutionary context. So you have to have variability in your population, it has to be heritable, and then the last thing that we need is some kind of selection. This is a box of chocolates, by the way, just to show selection. But we need selective pressure. So you need to have something that will cause some kind of difference in the survival of those offspring that have inherited that genetic variation. Okay? So you need to have some kind of environmental pressure. So it could be a climate change. As climate changes, those that have genes that might confer some kind of advantage might do better. It could be hunting and fishing pressure. Humans actually have influenced size of fish populations or horns on sheep because of hunting pressures. Or it could be toxic chemicals. So that's what I'm going to get to towards the end of this talk, is how we influence that process with the toxic chemicals. But I'm going to begin with how life evolved in a toxic world, so taking the very long view. So if we think about our planet, we were just talking about movies that have, science fiction movies, but, we're living in a science fiction movie. Earth is a pretty toxic place. If you landed here from another planet and hadn't, had this long evolutionary history here, this would be a nasty place. And so if we think about Earth and we think about the very beginnings, billions of years ago, when cells were just beginning to come together and forming on Earth, the conditions were really, we had a planet, we didn't have oxygen, right? There was no oxygen 3.8 billion years ago, 3.5 billion years ago when life was first taking shape. And without oxygen, there was no ozone layer. Without ozone layer, we all know that ultraviolet light can damage DNA, right? So ultraviolet light is very toxic, and there was less protection. So back on early Earth, ultraviolet light was incredibly intense, far more intense than it is now. And so if you think about early cells trying to survive, their DNA was at risk from ultraviolet light. If we move up a couple of billion years, Earth cells acquired the ability to photosynthesize and produce, and as a result, oxygen became a waste product. Okay, so back then, oxygen is a toxicant, right? Oxygen is toxic. People take antioxidants because you're worried about toxic oxygen radicals. Reactive oxygen can be very damaging to your cells. So oxygen was a problem, and it's said that back then that when cells started producing oxygen, that was one of the first pollutants produced by living things. Earth became polluted with oxygen from all of these little cells that started photosynthesizing and releasing oxygen as a waste product. So oxygen is toxic. And then if we move up billions of years, up to about 500 billion years ago, 500 million years ago, when terrestrial plants started colonizing the Earth. If you think about terrestrial plants were rooted, right? So they couldn't go anywhere. So one of the things that plants started doing for a defensive mechanism was that eventually they evolved nasty chemicals. Everybody knows plants can have plenty of toxic chemicals in them. We can't just go eat any plant because some of them are going to make us sick. So plants are very well known for producing lots of different alkaloids. We know some of those toxic chemicals as drugs. We harvest them for drugs. But plants produce a lot of really nasty chemicals. And so, to protect them against predators, whether it was animal predators, insect predators, so plants are pretty nasty. So it's a nasty place. But fortunately, evolution is amazing. And so if we go back and we think about, so one of the things I did, and these are things that I wrote about in that first book, so these two slides are really that whole first evolution in a toxic world in 2 slides. But we go back to the very origins. Some of the earliest cells on Earth had an enzyme called DNA photolysis, and it could repair. It was a DNA repair enzyme. And what it would do is repair the damage that ultraviolet light caused. And it's a highly conserved enzyme. It was probably present in some of those very early cells, protected them against ultraviolet light, and almost every living thing today still has an enzyme just like that. So it's called highly conserved. Just about every living thing except for placental mammals, which is a long story, which I won't go into, but it's really kind of amazing. So this was one of those enzymes that had evolved early. It was protective. There are other DNA repair enzymes, but DNA repair was very important back then with a lot of ultraviolet light. If we move up to oxygen, the time that when oxygen started flooding into the atmosphere, well, how did cells survive? because now all of a sudden we have this toxic oxygen. Back then, everything was anaerobic. It didn't like oxygen. Oxygen was bad. But now most of the things that on Earth, or animals, are aerobic, right? We actually need oxygen. How did this happen? So over time, life evolved enzymes that could detoxify oxygen. Catalase is one. Superoxide dismutase is another. So life evolved these enzymes that could detoxify oxygen, diffuse the toxicity caused by reactive oxygen species, and we still benefit from those today. When you go exercise, when you're just breathing, anything you do, you have these enzymes sort of in the background sopping up the reactive oxygen species that your cells are making as you breathe, as you just do anything. So these are really critical, highly conserved enzymes also. And then finally, in response to a lot of these plant toxicants, there's a whole family of enzymes that detoxify a lot of plant toxicants, a lot of plant poisons. They also are involved in a lot of drug metabolism because a lot of our drugs kind of come from plants. And so when I said that toxicologists tend to be very interested in metabolism, a lot of times they're studying these kinds of enzymes. It's a whole huge enzyme family. And here I just showed there's a really interesting story about Queen Anne's lace, the toxic chemicals that it produces, and this black swallowtail butterflies that can live off of those plants because they've evolved enzymes to detoxify the toxicants that the Queen Anne's lace makes. So, and we have lots of different kinds of these enzymes that we depend on for detoxifying, even things like they metabolize things that we take like Tylenol, alcohol, lots of things. So this is just an example of just 3 of those detoxification mechanisms. But the idea was to get people thinking about there are a lot of these naturally occurring toxicants, and over the course of evolution, millions of years, billions of years, hundreds of thousands of years, life evolved in response to these pressures caused by these naturally occurring toxic chemicals. And we have this amazing detoxification systems. But we all know that the problem today is that a lot of these systems have become overloaded, and they're being exposed to chemicals that they didn't evolve in response to. So here's a quote from Rachel Carson, which seemed appropriate today. The chemicals to which life is asked to make its adjustment are no longer merely the calcium and silica and copper and all the rest of the minerals washed out of the rocks and carried in the rivers to the sea. They are the synthetic creations of man's inventive mind, brewed in his laboratories and having no counterparts in nature. So back then, this was 62 when Rachel Carson said this, that, life evolved all these systems to basically detoxify chemicals, but what about today? So this is what I want to talk about next, and I'll spend the rest of the time talking about what about today in response to the chemicals that we produce today. So here we are. And as Yvonne mentioned, we have PCBs, we have lots of pesticides, herbicides, and even antibiotics is a toxicant to bacteria, right? So these are the kinds of things that I focused on for the second part and asked, can life evolve quickly in response to these kinds of chemicals? Because what we know today, what I just talked about, was sort of taking the long view and talking about evolution over long periods of time. But increasingly, evolutionary scientists are understanding that life can also evolve very rapidly. And we're not talking about the evolution in terms of the evolution of different species. We're not talking about the evolution of a whole new enzyme or a whole new protein, but we're talking about very small changes to some of these proteins and enzymes. It's still evolution because their whole population changes in response, but we're talking about small changes, and they're small changes, but they can have very big consequences. So this is the topic of that second book, which is Unnatural Selection, How We're Changing Life Gene by Gene. So I'm going to begin with antibiotics because I think this is something that everybody's familiar with, and if people haven't had their own experience with antibiotics and antibiotic resistance, they probably know somebody that does. It's a huge problem. I was just talking with Yvonne about this earlier, but antibiotic resistance, if you just have to read the news these days or listen to the radio, where you know that antibiotic resistance is increasingly becoming a big problem. And so each year in the US, and you probably might have even seen these numbers, I don't know how many times I've seen them when you just look in the news, but there's 2 million cases of antibiotic-resistant infections each year and 23,000 deaths in the US from antibiotic-resistant bacteria. So these were diseases that were once curable with antibiotics. How did we get here? So before I go into sort of the evolution part, I always think it's important to think about the chemicals that we're producing, why we're producing them, and how important they may or may not be to our lives. And so antibiotics, we all know, are really very important. These are some of the founders of very important scientists that were involved in this development and discovery of antibiotics. They were working in the early 1900s, late 1800s. Paul Ehrlich was one of the first who, just after the discovery that bacteria actually cause disease, he was working with the dye industry, and he wondered that if you could, at that time, you could actually stain bacteria and see them, because if you've ever done like cheek scrapings or tried to look at cells in a microscope, you know they're clear. They're very hard to distinguish from your own cells. But back then, someone had figured out that you could actually dye the cells and stain them, and then you could see them and distinguish them from human tissue. And so Ehrlich wondered, well, if you can dye cells and stain them and see them, could you poison the cells with a dye? So if you had some chemical that specifically sought out those bacterial cells, why not try to aim at them with that chemical and kill the cell with that chemical? So it was called a kind of magic bullet, aim chemically. And so Paul Ehrlich did come up with a dye that was effective at doing this. It was very specific, though, for syphilis, but it was a proof of concept. It showed that you could actually poison bacterial cell with a chemical. Following his work, Gerhard Domack and both Alexander Fleming, these were two scientists that had also served in World War I. And they saw many men die from their injuries that weren't, the injuries themselves weren't life-threatening, but the infections that followed were. And so these people were living in a time when people died of infections, and I think it's hard for all of us to realize this because it just doesn't happen anymore. But before antibiotics, people died from staph and strep and infections that we just think of today as curable. So Domack came back from World War I, and he was also determined to sort of pick up where Ehrlich left off and discovered dyes that could kill bacteria. He discovered sulfa drugs, which we still use today, back in the 1930s. But we probably most often think of Alexander Fleming. He working at the same time. His interest was antiseptics because he also saw the problems with infection and the diseases and the death and destruction that they caused. So he was looking at antiseptics, and his is the story of that accidental discovery that everybody might have heard of already. But Fleming was, he had plates full of bacteria. He was going off for vacation, so he left their agar plates that have sort of a lawn of bacteria on them. He left his plates for vacation, he went away, he came back, and he noticed that there was this mold growing on the plates, which isn't unusual. If anybody's ever worked with bacteria, you know that mold lands on plates and it grows. But what he did notice was that there was this clearing in the bacteria, and somehow that mold was secreting something that was killing the bacteria. And so he had the insight to follow up on that, and of course that was penicillin. So he discovered penicillin, and that became the kind of wonder drug, the miracle drug. And sulfa drugs and penicillin were very important during World War II for saving many lives, and then they became very popular antibiotics. And so Fleming won the Nobel Prize, along with two others that helped move penicillin from the laboratory into the market and make it in high enough concentrations that people could use it. But he realized early on that there could be a problem with using too much penicillin. And he said, I'd like to sound a note of warning. It is not difficult to make microbes resistant to penicillin in the laboratory by exposing them to concentrations not sufficient to kill them, and the same thing has occasionally happened in the body. So he knew that bacteria could evolve resistance, and he was worried about this. This was back in It was a long time ago, and apparently nobody listened, because here we are today with big concerns about antibiotic resistance, and it's not just to penicillin, it's to just about every antibiotic that we have. How does this happen? So if we go back to sort of the basics of evolution and we talk about genetic variation, where does variation come from? This is very basic, but we have when a cell divides, this is its DNA. This is a cell, it's DNA. DNA reproduces and replicates. So you get, these are, say, this is a strand of DNA. And the DNA opens up, it replicates, and you get two strands of DNA. These should be identical to each other. This should be identical, these two should be identical, but every once in a while a mistake happens. And that mistake is a mutation. Sometimes mutations are corrected and sometimes they're not. If they're not corrected, they just get carried through to the next cell, and then you end up having what's called a daughter cell with a mutation. Sometimes these mutations, actually most of the time, these mutations don't do anything. They're called neutral, they have no effect, they're carried through, no big deal. Sometimes they're lethal, and that cell won't go on anywhere. And very occasionally, they might be beneficial. And they can happen in bacterial populations. So now if we go back and we talk about mutation in bacterial populations, here we have a bacterial cell. Bacteria just divide in two, that's how they reproduce. So this bacteria divides in two, and a mutation might pop up, and it gets carried through the population. There's no mutation here, so we end up with a population of bacterial cells. Some of them have this mutation, some of them don't, no big deal. Bacteria, whether it's Staphylococcus or strep or whatever you might have, it's just doing fine, pumping away, reproducing, and so you get this genetic variation in the population. But maybe you don't feel so good, and so you go to the doctor, and the doctor gives you an antibiotic. And what happens is that the antibiotic just so happens that these cells with no mutation are sensitive to that antibiotic. But maybe that mutation made the cells resistant. And if it does, and you're exposed to antibiotics, so you've changed their environment, you've caused some selection pressure, and you end up with a population of cells that are resistant to that antibiotic. And then those cells go on. And if you pass them on to somebody else, that somebody else gets antibiotic-resistant staph or strep. This could be a mutation that happens in kind of real time while you're exposed to that. Or it could have been what people have discovered with bacteria, which is really kind of distressing, is it could have happened millions of years ago. So a lot of these bacteria tend to have resistance genes that must have evolved hundreds of thousands or millions of years ago. And if you think about it, it might make sense, because here we're talking about penicillin. A lot of our antibiotics are from living organisms, and they all live together in the soil. So it's possible that millions of years ago, they were all being exposed to some of these different chemicals, and they just happened to carry these resistance genes with them. This is called standing genetic variation. But one other thing about bacteria that makes them particularly tricky when it comes to resistance and concerning, so this is called vertical transmission of genetics. So what we do, we have vertical transmission. We have us and we have our offspring vertically. We don't give our genes to our sisters or to our friends, but bacteria can. So they can actually transfer their genes to other bacteria through these little bits of DNA called plasmids. And so they can transfer their resistance to bacteria that aren't related to them. And so this is one of the concerns about resistance, because when they talk about resistance in one kind of infectious disease, there's the potential that that resistance gene could be passed on to a different kind of pathogen. And this is part of the reason why there's so much concern about resistance being in just, when you hear about multi-drug resistant bacteria, you hear about bacteria that are becoming resistant to all antibiotics, and there's big concern not just for those pathogens, but because now those genes are kind of out there. So if we go back to the original thinking, how did they get resistant? What are the mechanisms? What changed in those bacteria that made them able to handle these new chemicals that are coming into their environment? They do all sorts of things. The detoxification enzymes can change just a bit so that they can now metabolize a new chemical. gene duplications, which is really interesting. So there can be, say, they can produce multiple copies of a gene that might be a beneficial gene. Some cells have pores that help pump out toxic chemicals. Well, what if they increase the number of those pores? Then they get better at pumping out toxic chemicals. Target site modifications. So a target site means when you make an antibiotic or a chemical that is going to kill something, right, you have a target and that chemical's going for that target. Well, if the target changes just a little bit and the chemical can no longer interact with that target, well, that organism now can resist that chemical. So there's a lot of different ways that living things can sort of resist and evolve resistance to toxic chemicals. And it goes back to sort of some of the basics. They're just sort of tweaking those basic systems that have evolved over longer periods of time. So what do we do? So I think that with antibiotics and just about everything else, everybody probably knows the answer because we hear about it a lot. But we can't do anything about genetic variation, especially with bacteria. It's already there. We can't do anything about the inheritance. You can't stop them from you can't stop them from passing their genes on if they reproduce. What we can do is lay off on the selection pressure. So there's a lot of talk about how do we change, how we, back when my kids were young, if I walked into the doctor's office they had an ear infection or a little temperature, I walked out with amoxicillin. That just doesn't happen, I don't think, so often anymore. Usually they want to know that it's a bacterial infection. With my kids, it could have been a viral infection, which antibiotics don't help against. So now they're much more careful about giving out antibiotics. There's a lot of new discoveries that are coming along. There's certain kinds of antibacterial chemicals that can kill very specific bacteria and not kill sort of all bacteria, because a lot of antibiotics are called broad spectrum or nonspecific. They don't kill all bacteria, but they kill a lot of different kinds of bacteria. And so that's a problem because you're exposing bacteria that don't need to be exposed to that chemical to antibiotics. So, but the biggest thing we can do is reduce the pressure, reduce that selection pressure. And there are studies that show that when you do back off, that the sensitive populations begin to emerge again. Because maybe there's a trade-off. Maybe the resistance genes also made them not so fit. So if you back off, sometimes the sensitivity comes back, and that's a good thing. Okay, so now I'm going to switch gears a little bit. So that was the sort of very fine end in moving on to weeds and herbicides in agriculture. Tell a similar story. So to begin with, what are we dealing with? So weeds are excellent subjects for the study of microevolution. That's what I'm talking about. Remember I said I'm not talking about evolution of new species or evolution of new proteins or enzymes. We're talking about little tweaks to proteins and enzymes, and we're talking about very small changes in quick time, and this is microevolution. And Herbert Baker was an evolutionary ecologist, and he also wrote about weeds, and what he knew about weeds, we know today too, is that weeds are really good subjects for this because they're very prolific. They produce lots of seeds. They might reproduce many times a year. They're not so picky about where they live. So weeds are weeds. They've got a lot going on, and they tend to be very diverse, and they have the potential for a lot of genetic diversity. So weeds are sort of powerful little things. And then if we think about agriculture, what have we done? This is E.O. Wilson in 1993. Artificial selection has always been a trade-off between genetic creation of traits desired by humans and an unintended but inevitable weakness in the face of natural enemies. So when we breed crops for the traits that we want, the characteristics we want, we've basically turned them into pets. They need care. They can't really care for themselves. And we also, as I was talking about Yvonne earlier, she was mentioning monoculture. We plant very large swaths of these crops, which just kind of invites attack by pests. So we've created this system where we need to help out our agricultural crops, and we do this with herbicides. There are at least 100 different kinds of herbicides. But that's not enough. This is one of the herbicides that I'm going to talk about is Roundup or glyphosate. So that's also been in the news a lot lately. The I can't remember what it's called, IARC just came out and said that Roundup is, I think, a probable carcinogen. So, but it had been considered one of the fairly kind of safer herbicides for many years. And so Monsanto sells billions of dollars a year of pesticides and mostly Roundup. So it's big business. How did we get here? How did Roundup become this very popular herbicide? It was discovered back in the maybe the 70s, by John Franz, and it turned out it was a really effective herbicide, and it was effective because it basically kills anything green and growing. And so what it allowed farmers to do was to just kind of burn down crop. It helped create the no-till agriculture. So because if you could just burn down all your crops with the herbicide, or all the weeds and the crops before you plant, then it pretty much went away. Then you could plant again, and you wouldn't have to worry about tilling and disturbing the soil. So in that sense, it was considered a good thing. And at around the same time, or a little bit later, Paul Berg is a pioneer of biotechnology. He won the Nobel Prize for some of the early work in genetic transforming genes from one organism to another. And so what Monsanto thought was, well, what if we have a herbicide that is incredibly effective? Well, what if we could make a plant that could grow and be resistant to that herbicide? So then we could apply it even when the plant was growing. It would kill everything else. We won't have to have any weeds, but the plant will do just fine. It took them many years, but they eventually did it. And Roundup-ready soybeans were developed and produced in, I believe, the 90s. That was the first Roundup-ready crop. They also produced corn, cotton, canola, alfalfa, and beets. So this is to say that there were a lot of Roundup-ready crops that were being produced. One of the special things, and one of the reasons they did this, was because they thought that it would be very difficult for anything to evolve resistance to Roundup. It took them a long time to develop their own Roundup-ready crops and to find a gene that could be implanted into plants and that could confer resistance, and it was very hard to find this. And so the thought was it just wouldn't happen in nature. It would be hard to for that to happen. But once they started producing all of these Roundup-ready crops, think about that selection, the box of chocolates, and the environmental pressures. It created an incredible pressure on the weeds because, so back in 1996, before these crops were everywhere, they sold 180 million pounds of Roundup. They're now projected to be selling in the millions of metrics of tons of Roundup. Okay, so that pressure increased incredibly as farmers started using Roundup after Roundup after Roundup on their crops. And what did we get? We got Roundup-resistant weeds all over the place. This is a map from Dr. Kevin Bradley from the University of Missouri. This is 2013, and it would probably look different now. But it just shows the number of different kinds of weeds that are agriculturally important that are resistant to Roundup. Roundup resistance is a really big problem for agriculture. They've got these Roundup-ready crops, so they're using the Roundup, and now they've got Roundup-ready, Roundup-resistant weeds. It's not just Roundup. Like I said, there's 100 different herbicides. There's 200 different herbicide-resistant weed species. And 60 million farms across the U.S. have problems with resistant weeds. So that's a big deal. And again, solution, for the ever since we had pesticides and herbicides, we've been at war with nature, and we've been relying on chemicals to beat down the pests and the weeds. And we're now realizing that this isn't the best solution because nature wins. Weeds evolve resistance. And so we need to figure out a way to turn down the pressure, to use less of the herbicides, and to figure out how to farm that way. The last thing I want to talk about is one of my favorite subjects, which is bedbugs. So I usually ask if anybody's had bedbug problems in the audience. And usually when I do this, it's the young people, the college students and the graduate students that have had bedbug issues, and the people my age haven't, because I grew up in kind of a bedbug-free era. And we all know that bedbugs are making a resurgence. There's actually a new book out about bedbug resurgence, but bedbugs have come back, and they're, of course, resistant to some of the pesticides that we use. How did this happen? Well, we can go back to the early days. We have Paul Mueller, who discovered DDT, or he didn't discover it, he discovered that it's a very good pesticide. And what he found was that it was incredibly good at killing houseflies. DDT became the go-to chemical. DDT was sprayed everywhere and anywhere. For the book, I interviewed Claude Boyd, who was an ecology graduate student back in the 60s. And he said, that there was a culture of using pesticides. Everybody knew they were probably using too much, but nobody wanted to stop. He was in Mississippi. Mosquitoes were a big problem back then, and DDT was a savior. He said, they would spray it in their house. They would go out, get some air, let it settle, and come back in. There was a radio station, WDDT, so it was a big deal. And by 1973, over 1 billion pounds of DDT were used. And of course, this is where Rachel Carson, really wrote about, was the DDT and the environmental problems caused by DDT. She did mention resistance, but beyond resistance, the problem was that it accumulated, it didn't go away, and it caused problems for raptors and other animals. But it's not just DDT. If this is a chart from a book about evolution, it's a very good textbook by Emery and Carl Zimmer, Doug Emery and Carl Zimmer, and this is just showing resistance to pesticides in houseflies over the years. And as you can see, they're pretty good at resisting pesticides. There's a lot of different pesticides here. This arrow is when resistance was first detected, and this is when the housefly population was pretty much considered resistant, and the chemical wasn't useful against them. Okay, so here's DDT, but every other chemical that's been used, we have a resistance problem. Here's Rachel Carson again. Darwin himself could scarcely have found a better example of the operation of natural selection. Again, years ago, decades ago, she was talking about this, yet we continued. Nobody was talking about this in terms of, what do we do? What should we do? How do we stop this from happening? And again, the answer is the same as always. We need to turn down the pressure. So I talked about just bacteria, weeds, and insects. This is across the spectrum of life. I could have talked about viruses and evolution in response to antivirals, cancer cells in response to chemotherapy, fish, fish and birds and salamanders. These are evolving in response to pollution, to chemicals like DDT and PCBs and mercury. This is kind of amazing. That's sort of like evolution out in the wild. So if you have questions about that, I can answer that afterwards. And then there are questions about whether humans are evolving. Most likely, so the problem is, as you can see, is that those species that are evolving in response to the toxic chemicals and are surviving are those that are like weeds, bacteria, insects, things that are very prolific, short lifespans, evolve rapidly. The fish that are evolving are similar to that. They have short lifespans. They are pretty prolific. So it's those of us who live longer, have longer generation times. We're kind of out of luck. So just the take-home message here is that we influence evolutionary processes with our chemicals, and that evolution is really relevant in our everyday lives. I think we just don't usually think like that, but evolution can impact us in big ways that we don't always think about. So that's it, and I'd just like to say thank you to, first of all, Island Press, which is, I've written the books with Island Press. It's a great press to work with, and they're very supportive of sort of books that make a difference, and they really have been great to work with. So this is the new book. This is the Rapid Evolution, and sort of a book written for the general public and general readership. This was more written for toxicologists, Evolution and Toxic World. That was the first 2 slides that I showed, that sort of why we should think about this. Then I need to thank my husband and my friend Penny, because they were always, my friend Penny was my AP biology buddy, And she is now an immunologist, and she has read every single page that I've written and reviewed it as a scientist. So valuable. My husband, who's also a scientist and has read much of what I wrote, and my dad, who always encouraged me to write. And I always said, no, scientists don't write. So I didn't start writing until I started doing these books, which is only a few years ago. And then the UMass Department of Environmental Conservation, where I'm an adjunct, but for the access to their library, which without the library access in their database, I wouldn't be able to do this. So that's it. Thank you. Anybody have any questions?
Our bodies are made up of tons of bacteria.
Mm-hmm.
So would that help us evolve to deal with these?
Well, that's an interesting question. I haven't thought about it in that way. Well, you mean evolve in response to other chemicals?
Toxins, yeah. Learning how to deal with them.
That's a really interesting question, because there are, I would probably say, I doubt it, but I don't know. I don't know if anybody knows. So bacteria are in the environment. There are lots of examples where bacteria have evolved resistance or even turn nasty stuff into food sources. That's actually, I think, when Monsanto discovered the gene for glyphosate resistance or Roundup resistance, they were hoping to find bacteria, and they found it in a bacterium that was in waste ponds that could really do this, waste ponds that had Roundup in it. And they were hoping to find a gene in bacteria that could degrade the stuff and clean it up. So there's a lot of examples of that. So I never really thought about it, like in our bodies, would you have that?
Because I think we're more bacterial cells than we are human cells.
We are. We are. I don't, but you have to think about that absorption, distribution, excretion, that kind of thing. And so those bacteria that are out in nature that evolve resistance and things, to mercury or even to PCBs, or that can, detoxify those are constantly exposed to those kinds of chemicals. And so over years, generations have become resistant. It's a very interesting question. My guess would probably be not. But I don't know. And usually, often when bacteria are evolving resistance, those chemicals are in really high concentrations. So in the places that they degrade chemical toxicants in the environment, often it's where the chemicals are hotspots. So they're actually, I might say this wrong, but I think in some places, like there might be bacteria that could, say, metabolize PCBs and help change PCBs and detoxify, not necessarily detoxify them, but alter them. But it tends to be where PCBs are really high. So we thankfully don't have that situation in our bodies, even though we are exposed to lots of different chemicals. So, but it's an interesting question. And of course, that is another downside of antibiotics, is that we're flushing out a lot of the beneficial bacteria, and nobody really fully understood that until only recently. So that's another reason to back off of antibiotics and to think about how we could treat disease differently if we think about it more ecologically. And it's the same thing out in the farm fields too. We need to think about things sort of more ecologically in the whole system rather than just taking care of one kind of crop. Yeah?
A recent article, and I think Nutrition Action, covered toxins in fish, and one of the striking things was that fish that were spawned in farms were more dangerous to eat than wild-caught fish. And it was stemmed from, I believe, the food that was given to them. Could you talk about the process of that, how it gets into that food supply, and then how does the body take care of those toxins, like whatever mercury or PCBs or whatever in the fish?
Okay. Yeah, so I didn't know that was still a problem. I thought that they had already... I mean, years ago, I guess I had heard about... So, one of the problems is often they're feeding fish. So they're taking fish from the wild, and they're feeding the farmed fish, if that's how they got that way. And so then it's your bioaccumulation, biomagnification problem. So you're kind of... And sometimes they're feeding them fish oil, or they might be trying to, concentrate it. And a lot of the chemicals that tend to be the problem chemicals, like the organochlorines, like the PCBs and the DDTs, even though they're called legacy chemicals because we don't produce them and release them, well, not here anyway, anymore, those concentrate in the fatty tissue and in the fats. So in oil would be the worst thing for those kinds of chemicals. Other chemicals tend to be in the muscle. So anyway, they're feeding, I thought that now that they would test the fish fodder that they're feeding to agriculturally grown fish or aquaculture. But if it's the case, that's how I think it probably gets there. If it's from the food, it's because they're taking fish from the wild. Those populations tend to have chemicals in them still. And even if they're small fish, if you feed enough of small fish, they can accumulate sufficient concentrations to where they're a problem. So what happens to us? It depends on the chemical. But I think there's plenty of studies that show that we have lots of different kinds of chemicals in us, including some of the ones that little small concentrations of DDTs and other kinds of chemicals that you would be surprised that we have in us. And it could be from just being exposed through either eating, fish and plants that might still be exposed to those in some way. So, and, they'll eventually, PCBs, DDTs, they do accumulate in fat, so they stick around. One of the problems with these chemicals, which people, especially maybe in the AP bio, you might know this, and other people probably know too, but, they get in your fat. One of the problems with this is that they travel in the fat, and so they get, one way that they get out of a body is they go out with the fat. So when a woman is lactating, milk is fatty, these chemicals go out in the fat. And for wild animals, it's a problem, especially oviparous animals that produce eggs. Eggs are very fatty, so these chemicals go in the fat. Female fish tend to sometimes, once they spawn, because they spawn a lot of little fatty eggs, so they're basically dumping all their organochlorine or fat-accumulating chemicals into their eggs. And so they'll reduce their own load of those chemicals, and it will come out into the eggs. And so then the most vulnerable population, the young fish and young kids, if we're talking about lactation, are exposed to the chemicals.
Yeah.
Yes?
A few years ago, I worked in cell bioremediation, how to clean oil by bioremediation, not by chemical. When I made some engineering work around the refining and oil, I thought, okay, this is chemical, it has many consequences, healthy body, environment, and this is great product. This is bacteria. He said, oops, bacteria, this is terrible thing. Because the knowledge is limited.
Yeah.
Do you think one of the most important challenge is concept?
Yep.
Bacteria still around the negative thing.
Oh yeah. Yeah, and I think it's changing now with all, and it's actually, this is one thing that bugs me. We tend to just be black or white. We go to all in one way or all the other. So now there's all this stuff about the microbiome, and now they're like making the microbiome is all the bacteria on your body and in your body, and some of it's beneficial, a lot of it's probably beneficial, or just sitting there and not doing anything and not pathogenic. And now everybody's like, you can get a microbiome spray, you can do all this stuff. So, but somewhere in the middle, yeah. So even, it's true. So we're starting now to think, oh, maybe bacteria aren't that bad. And it will be the same thing that, oh, so bacteria in the environment, maybe they're not that bad. Because people don't realize that the environment's loaded with bacteria and always has been. Not just bacteria, but fungus and viruses and protozoa and all sorts of little things we can't see. And the world's crawling with it. And we've lived with it. But we're there's all this new thing about how we're over-hygienic. And we're just too clean, and we just do too many things that, are affecting just our own microbiome. So, but I think in the environment too, people are squeamish. We're like over squeamish about these kinds of things. But in bioremediation, it's a big deal. I mean, bacteria are great for cleaning up a lot of the messes. So they're important and they're useful. And I think there's a pretty big business though of trying to get bacteria, that are able to clean up a lot of the toxic waste sites. Or at least reduce the hazard, not completely clean up. So, but that's a good point. Yeah, it's concept. It's just how we think about these things. So we need to change. But I think that's happening now. It's just that right now we're like all on we all, bacteria are great and they'll do everything and everything is caused by bacteria. And we'll probably find out how important they really are. But, somewhere there needs to be some moderation. So, anything else? Well, yeah.
You talked about Roundup as the herbicide Roundup, but the relationship between the Roundup and the Monsanto that produces Roundup and produces Roundup-ready seeds and monopolizes the market and creates the conditions for monoculture and is powerful in terms of its lobbying ability to suppress or defend against the diversification that is needed to preserve the possibility of agricultural crops in the future. They think in terms of quarterly profits and generations. And that, to me, is a really frightening thing. And it's kind of tainted the idea of genetic modification of seeds.
Yep.
People think of that as an inherent evil.
Right.
Kind of like vaccines are no good anymore.
Right.
And the technology might be very powerful and useful if it were used in a socially responsible way as opposed to a corporately profitable way.
Yeah, that's a really good point. Actually, my next project is sort of thinking about the response to all of this. So what do we do? And one of the things that I want to learn about more and write about is that concept of, if the big users of GMOs had been, as you say, socially responsible, all for the good, would we think about them differently? I think there would still be people who are concerned about them, but I think it wouldn't be the same pushback as there is when we've got these, this kind of cropping system, and then they produce more cropping systems. And I think even some of the early scientists that might have supported the use of Roundup, they supported it with the notion that there'd be crop rotations with things that didn't use Roundup, but it didn't happen that way. So, I totally agree, and I think that's a really interesting point, and it's something that I'm looking forward to really digging into.
The interaction of these different chemicals that get into natural systems and the effect maybe of chemical A on, let's say, frogs, which have very thin skin, so they tend to absorb chemicals more easily than other organisms.
Mm-hmm.
But how do we know what the results of the interaction of the chemicals are on the frogs or on the salamanders? That
is one of the biggest questions. So years ago I wrote a paper on sort of the evolution of toxicology as a field, because the way toxicology as a field evolved is studying one chemical in one system at a time. And it evolved in that way because it's really a regulatory and applied field. And so the regulators needed numbers, and they needed to control these single chemicals. And the part that got left out is the realization that we're exposed to lots of different chemicals. So whenever there's like water quality criteria, it's for a single chemical. It's not for the 20 different chemicals. So what happens when those 20 different chemicals are all below the water quality criteria, but you have a lot of them? So to make up for that, they're trying to do, there's sort of models to how do you think about this. There are new systems, they're called high throughput systems. Can you sort of use very small amounts of chemicals, look at them in cell systems, run them through a computer so that you could do lots of different combinations of chemicals, but still that's at the cellular level. So it's a really big problem in toxicology. And even when people do these studies in a laboratory and in laboratory animals, they can only really look at a couple of combinations of chemicals because you're talking about enormous systems to do that. So it's a big deal in sort of, people trying to do modeling and computational toxicology and all sorts of things. And so that's another question, sort of like the GMO one, that I would like, if toxicology had evolved or came about it in a different way, sort of in response to, say, maybe chemical mixtures, how would the science look? What would it look like as a science? It would be very different, I think. And so, but it didn't. And we can't start over, so we need to rethink how we do things. Or some things probably do need to start over. But that's a good question, and it's a big problem because, even I slip into that sort of talking about here all this, single chemicals, and really we're talking about chemical mixtures. So, and when you're talking about resistance in chemical mixtures, that's actually one of the solutions. When people talk about chemotherapy, often there's chemical cocktails, antibiotic mixture, so there's just sort of, if you throw a couple of different chemicals at a system, it's less likely that they would have resistance to those multiple chemicals. So, and the same thing with pesticides and herbicides. There's even talk about, and I think it's in the process if it hasn't been approved already, I don't know where it's at, but they call it stacking, so you don't have just one, a plant resistant to just one herbicide, you have them resistant to 2 herbicides, so that so now you can beat off the resistant weeds by applying 2 different herbicides. So if it's resistant to Roundup, you throw another herbicide at it, and the plants can withstand it because you've engineered in resistance to both of those, which seems like it will just create a bigger problem of more herbicide use, and eventually, I would guess, resistance will happen. So.
Isn't herbicide usage increased like 15-fold?
I don't know what the fold is, but it's increased a lot. But I don't know what the fold is. It probably depends too what herbicide you're thinking of, what you're talking about. And one of the problems also with like glyphosate, well, now that glyphosate is, people are sort of rethinking it as the quote safest herbicide, but now that there's so much resistance to it, some of the older herbicides that fell by the wayside because they were more toxic are coming back into use because I think they weren't creating lots of different herbicides because they had Roundup. So the development what's one of the problems with antibiotics too is that the development of antibiotics is sort of stunted back in the, there haven't been a whole lot of new antibiotics, very new, totally different kinds, not just tweaks on the old kind. That have been being produced. So that was why it was big news a little while ago when, I can't remember his name, but from Northeastern, discovered a new, soil microbe that produces an antibiotic. You had Kim, what's his name? Dr.
Yes, Kim Lewis.
Kim Lewis. So, that's a big deal because it's rare. For as long as it lasts. So, but now hopefully we'd be smarter about it. I think he thinks
It's a unique structure.
Right.
It's not just the bacterium, but the method. It's very interesting.
Yeah, and so there's some, I guess the talk was that it would be difficult to evolve resistance, but, I feel like we've seen this over and over again. They said that with Roundup, so better safe than sorry. Hopefully we learned our lesson. We didn't learn it 45 years, 75, what was it, 65? We didn't learn it in 1945. Yeah.
Do you think there could be an evolutionary component in like the increased incidence of peanut allergies, for example? I don't know what brings about peanut allergies, but there's clearly an increased incidence, and I don't know whether anybody's seen whether is that passed on in any way?
I haven't seen that. It would probably be the reverse because it's a bad thing to have. But I did a little bit of writing about it a few years ago, so my information on peanut allergies is a few years old. But I think the thinking is part of it could be the way we process peanuts. It could be that we withheld peanuts. I mean, there's some research showing that exposing young kids to very small amounts, prevented, or that there was a lower incidence of peanut allergies. So I don't know where the research is now, but I think some of it is more just our behavior around peanuts and what we do with peanuts and how that might have contributed to that. But there could be other things. I'm not up on it, but I just know that we have changed. There's the whole, we're very clean, so we've changed a lot of things.
Some people have said that may be why there's a higher incidence of asthma, that people don't play in the dirt enough.
Yeah, there's all sorts of interesting studies that, I don't know if they're just one-off studies and the next study might show something different. So that's where it's hard to just say, oh yeah, and there's this study, because there's the one with the dishwasher, people who don't have dishwashers had I don't know what it was, less allergies or something like that too. So they were exposed to more stuff. And so, you build up a different kind of system. So I don't know, because a lot of this research is really new. And that's one of the problems is, I think, in science is that, especially these days and in writing about science, there's a lot of hype. In every article that comes out, there's a lot of hype because it's a screaming headline. And then 2 years later, maybe it flip-flops or it wasn't that big a deal. And then people kind of distrust. It kind of builds distrust of science, which is sort of a problem. Yeah, thank you. You're a great audience.